Hi, gang. How are you doing today?
Doing well. How about yourself?
Doing okay, thanks. Welcome back, everybody. So, okay, I'm gonna get started here. And so let me just talk a little bit about the syllabus, okay? So today is November 11th. Happy Veterans Day for anybody that served our nation. We are going to be wrapping up coverage of Chapter 5 today, and I'm hoping I can at least start a little bit of Chapter 6. We'll see how that goes. At any rate, this coming Tuesday, November 16th, this coming Tuesday, the 16th, I'll be assigning an additional homework assignment out of your workbook. Okay, so I'm kind of just telling you that's in your syllabus. And so if you want to get started on that a little bit early over the weekend, you know, if you appreciate just having a little bit more time to wrestle with sort of math topics, you might want to do that. And so the homework assignment is going to be workbook assignment number 4. Let me show you what that looks like. Okay, so hold on a sec. No. Yeah, this is homework assignment number 4 in the workbook. Okay, the same workbook you did the unit conversion, same workbook you did the graph interpretation questions a little bit ago. So I have a math word problem here that I want you to wrestle with. So again, I'm not assigning it today, but it is going to be assigned next Tuesday. So if you prefer to just start wrestling with it early, that's quite okay. So that's workbook assignment number 4. If you want a little bit of extra time on that. And I will say to you, to complete workbook assignment number 4, you're going to need some mathematical equations, some formulas, which I will introduce you to in chapter 6. However, I'll also mention in the chat window that if you want to get a head start on these things in your textbook, have a look at appendix B as in boy. Take a look at appendix B as in boy in your textbook, and you will find these math equations that I'm talking about that you'll need to do your workbook assignment number 4. Okay, so just a little gentle forewarning that this is coming up next week. You don't have to deal with it this weekend if you don't want to. But if you want to, there you know what you need to do, okay? All right. Okay, folks. So any questions about our schedule, right? We're heading into the bottom third of our semester together, so very quickly things are going to wrap up. So we want to stay organized and as focused as we can. All right. Yasmin, I see your comment to me in the chat window. I definitely understand. And if I call on you today by accident, just remind me, okay? So I hope you're feeling okay. All right. So let's continue, folks. All right. So from last time, we started talking about glycolysis. And as I mentioned to you previously, I think the best way to go about understanding this is not to have me just lecture on it, because then you take notes, but you don't process, and I need you to process. So I'm going to step you through these processes. You know, I already showed you a video last time about aerobic cellular respiration. Okay, so I'm going to step you through these processes with a series of questions to help stimulate your thinking so that when you're studying these things, these topics are making more sense to you. Okay, so let's just quickly read and answer the questions we've already done just to get our cerebral juices flowing, so to speak, right? The first question was, and I'm looking at figure 5.12 in your textbook, figure 5.12. Please have that open and available at this time. What are the names of the substrates at chemical reaction number 1? And the answers were glucose and ATP. So the purpose of that question is so that you understand what a substrate is. A substrate is any molecule modified by the enzyme. Second question was, what are the names of the products at chemical reaction number 1? The products are going to be ADP and glucose 6-phosphate. So what is a product? Any molecule made from the activity of an enzyme. Question 3, what is the name of the substrate at chemical reaction number 2? The substrate at chemical reaction number 2 is glucose 6-phosphate. Okay, the purpose of this question was to highlight this important principle. Notice that in biochemical pathways, right, glycolysis is a biochemical pathway. It's a group of chemical reactions that happen in a linear fashion. The first reaction happens, then the second reaction happens, then the third reaction happens. It's like knocking down a row of dominoes. You knock the first one down, then the second one drops, then the third one drops, then the fourth one drops, all right? So in biochemical pathways, what's important to realize is that the product of one reaction becomes the substrate for the next reaction. Just like knocking down the previous domino will cause the forward domino to fall, okay? Question 4, what is present at each orange ball but not actually printed in the textbook? And the answer is an enzyme. You must appreciate that each chemical reaction here is being made possible by a specific enzyme, which is not being printed in your textbook in figure 512. And honestly, I won't expect you to memorize any of these things, but I expect you to be able to interpret them when presented with information. Okay, any questions so far? Okay, so gang, let's see here. I'm going to continue my my conversation where I give folks questions. Okay, so I know I've given some of you questions and others not questions. So Alexis Pearson, did I give you a question?
Yes.
Okay, thank you, Alexis. Alicia, did I give you a question?
Yeah.
I did. Okay, so Brianna, I don't think I gave you a question. Brianna, did I give you a question?
No.
Okay, you sound so like, oh my God, no. Okay, fine. All right, so Brianna, relax, smile. This is a happy, friendly place. Okay, so I'm going to ask you a question, Brianna. So in figure 512, Brianna, is chemical reaction number 1 a catabolic reaction or an anabolic reaction?
Give me 1 second to look at it.
Okay. All right. So I'm just going to read the question again as Brianna gets a chance to get her thoughts together. So in figure 5.12, is chemical reaction number 1 a catabolic reaction or an anabolic reaction? What do you think, Brianna?
Is it anabolic reaction?
Okay, so why did you say that?
I don't know, because I didn't think it was catabolic.
Okay, that was an honest answer, and I appreciate it. Brianna, it turns out you are right and wrong at the same time, and by that silly little response I'm giving you, I'm telling you chemical reaction number 1 is both a catabolic reaction and an anabolic reaction. Let's break that down. Thanks for trying today, Brianna. I appreciate it. Okay, both. Okay, so let's take a look at this. Okay, an anabolic reaction is one in which we synthesize something that hasn't existed before, right? Think about anabolic steroids. We build things, we make things. So in chemical reaction 1 there, isn't it true that at the end of chemical reaction 1, we have glucose 6-phosphate? So glucose 6-phosphate is made, which means chemical reaction number 1 is an anabolic reaction from that perspective. Okay. Okay. Now, if we take a look at the side reaction there, we notice in order to manufacture glucose 6-phosphate, we actually have to break down ATP, don't we? On the left side of chemical reaction number 1, there's a side arrow where it shows ATP is being converted to ADP. So we're taking a phosphate atom and we are ripping it off ATP so I can link it on to glucose. So that means that ATP is being broken down, so that's catabolism, right? So chemical reaction number 1 there is kind of difficult to interpret because there's actually 2 things going on, and both things are made possible by the same enzyme. The same enzyme is grabbing onto, so if my hand is the enzyme, my red pen could be glucose and my blue pen could be ATP. So the enzyme is grabbing onto 2 molecules, which are effectively both substrates at that moment, ATP, which is the substrate, and glucose, which is the substrate, and doing some chemical gymnastics to rip a phosphate atom off the ATP and stick it on the glucose. So is chemical reaction number 1 anabolic or catabolic? The answer is it's both, because 2 things are happening simultaneously. Okay. Because ATP is broken down, this reaction is also catabolic. Okay. Folks, question to you in the chat window. Am I making sense? Okay, is there anything that you'd like to ask about this? Please feel free to ask. That's why we're here. Okay. All right, let's continue then. Next question. All right. Okay, let's see what to do next, ain't. Okay. All right, so the next question will go to Chelsea. Okay, Chelsea, you're up to bat here. Okay, so Chelsea, Chelsea, are you with me? Chelsea, can you unmute your microphone and say hi? Okay, Chelsea, you're having a technical issue, I guess. Okay. All right, Chelsea, I'm going to move on from you, and I'll come back when maybe you can solve that technical issue. Okay. All right. Okay. So Chelsea, you're receiving a message that I'm not allowing you to unmute. That's not true. I'm not doing that. Let's see here. Okay, Chelsea, I can't explain that, so we'll just move on for the time being. Okay, see if you can wrestle with anything there. It might just be something where we have to reset the computer. Hang in there. Okay, so next, Elena, I'm going to go with you next. Elena, are you able to unmute?
Yes, can you hear me?
I can. Thanks, Elena. Okay, Elena. So Elena, let me ask you this. In figure 512, do you see chemical reaction number 4?
Yes.
Okay, so here's the deal. When you look at chemical reaction number 4, Elena, you notice it starts with fructose 1,6-diphosphate, and then you can get one of 2 possible outcomes. You can get dihydroxyacetone phosphate on the left or glyceraldehyde 3-phosphate on the right. Either of those outcomes is possible here. But here's my question to you, Elena. At chemical reaction 4, when fructose 1,6-diphosphate goes to either of the 3 carbon molecules, is fructose 1,6-diphosphate being oxidized or reduced?
It's being oxidized.
Okay, that's a correct answer. Can you explain to us how you knew that was right?
How can I explain this? So when you're oxidizing, you're breaking down, and when you're reducing, you're building.
Beautifully done, Elena. Well done. Good job. Thanks for participating today. You did great.
Mhm.
Okay, absolutely. So the answer is oxidized. And what Elena said there in terms of explanation is super important. Okay, so many of the chemical reactions that you're going to encounter in aerobic cellular respiration, and honestly, all the chemistry of metabolism, involve oxidation and reduction principles. Okay, so let's define these terms. Okay, when a molecule is oxidized, when a molecule is oxidized, you break chemical bonds. Okay, you break chemical bonds. Elena was absolutely right about that. And what I want to point out and what is going to become apparent a little bit later on is when you oxidize molecules, you break bonds. And when you break bonds, you release electrons that can be used in other chemical reactions. Okay. When you break chemical bonds, electrons are released that can be used in other chemical reactions. Okay. Now, when a molecule is reduced, you actually create a chemical bond. And what I'll say with this is that for a chemical bond to be created, you need to provide a source of electrons to be part of that bond. Well, where are you going to get these electrons? Typically, you get it from another molecule that was just broken down, right? So you can see this interrelationship between oxidation and reduction. They kind of go together like peanut butter and jelly. They kind of go together like two sides of a coin. When you oxidize one molecule, you rip electrons off that then can be injected in another molecule to create new chemical bonds. Okay. Okay, nicely done. Very good. All right. Okay. All right. Any questions so far? Okay, let's continue. Okay. Oh boy, I gotta watch my time here. It's good to go through things this way, but it's so time consuming. Okay. Let's see here. Erica Jones. Hi, Erica. Can you say hi to us, please?
Hi.
Good afternoon, Erica. You doing okay?
Yeah, I'm doing all right.
Okay. All right. So here's my question for you, Erica. And so let me take a look at my chemical pathway here. All right. So Erica, do you see chemical reaction number 6 there?
Yes.
Okay, so I'm looking at the reaction where you see glyceraldehyde 3-phosphate going to 1,3-diphosphoglyceric acid, and to the left of that chemical reaction number 6, you have a circular arrow which shows 2 NAD plus going to 2 NADH. Okay, so Erica, here's my question for you. What does NAD stand for?
Um, I'm not sure.
Okay, Erica, I'm going to answer this one for you, and then I'm going to give you another one, okay? So NAD stands for nicotinamide adenine dinucleotide. He's like, yeah, okay, I was ever going to get that, right? Okay. You will get it someday. I mean, you know, if you have to. Okay, Erica, my next question to you is, if you look at chemical reaction 6, you see 2 NAD plus. Okay, so here's the question, Erica. Is NAD plus in an oxidized or reduced state? Is NAD plus in an oxidized or reduced state?
Oxidized?
How do you know that?
Because is it because it's being formed in the NADH?
No, that's not it, Erica, but let me see if I can draw this answer out of you, because I think you know the answer even if you don't realize it. So Erica, let me ask you this. If you have a molecule where there's no plus or minus, does that mean the molecule is electrically neutral?
Yes.
You are correct. That's what it means. So NAD does have a plus sign in this equation and in this question. So that means that NAD has an excess of positive charge. What would cause a molecule to have an excess of positive charge? What do you think, Erica? Okay. Erica, let me feed you a question and see, and just give you an option to answer it. Erica, is NAD positively charged because it has lost electrons?
No.
Ugh, okay, good attempt, but the answer is yes, Erica. Okay, the answer is yes. Okay, thanks, Erica. I appreciate it. And if you have questions about this, feel free to ask them. I appreciate your attempts to answer today. I really do. So, folks, NAD plus is in an oxidized state. How do we know this? Because NAD plus has a plus charge. That means it has more positive charge than negative charge. Because if it had equal positive and negative charge, it would be neutral. Since it's not neutral, because it says it's got a positive charge, that means it has more positive charge than negative, which means it has lost electrons. What satisfies the condition where we lose electrons? Oxidation, because we just defined it up here. When a molecule is oxidized, you break electrons. Excuse me. When a molecule is oxidized, you break chemical bonds. When you break chemical bonds, electrons are released. So NAD plus has an excess of positive charge because at some moment before it became NAD plus, it lost electrons. Some kind of bond was broken on this molecule, and in the next split second, electrons zipped off the molecule, went someplace else to do other chemistry, and the molecule we started with takes on an excess positive charge. So NAD plus is oxidized because this molecule has a positive charge, meaning the molecule lost electrons and can no longer neutralize the excess positive charge, which you know is due to protons in the nucleus. Okay, these aren't the easiest questions sometimes, are they? But folks, are you okay? Anything you'd like to ask? Are you passed out? All right, okay. All right, folks. So, okay, let's keep going here. Okay, rather than asking some more questions about this, I'm going to give you some information about NAD plus, or about NAD. Okay, so NAD plus, I'll say it this way: when NAD plus accepts electrons from another source, you can add hydrogen atoms to the molecule to become NADH. Do you see that at chemical reaction 6? NAD plus goes to NADH, right? NAD plus goes to NADH. So when NAD plus accepts electrons from another source, what that other source could be any other molecule that just lost electrons. Okay. So you can take those electrons that another molecule lost, and you can combine them with a hydrogen ion that's floating around the environment, and you can glue them to NAD plus to build NADH. Okay. So here's the deal: NADH is an electron carrying molecule. It's an electron carrier, okay? It's an electron carrier. Hopefully that's come out of some of the reading that you have done. Okay, take a look at figure 5.10. Take a look at figure 5.10 in your textbook. Okay, figure 5.10 would be relevant here, okay? Examine figure 5.10 and notice, if I read that chemical equation starting on the left, we start with an organic molecule. We have NAD plus, which has been part of our conversation. We go to the right, we have an oxidized molecule. The molecule that just lost electrons injects electrons into the structure of NAD plus, and we end up with NADH on the right side of the chemical equation. Okay. Folks, let me show you a video on the NAD plus system. Here we go. Let me get this linked up here for you. There we go. All right, folks, let's watch this. And here we go.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen...
Folks, think about it. To remove that hydrogen from that molecule, you had to break a bond. To break that bond means you released electrons
from a molecule. Since a hydrogen consists of a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all
So in other words, the product is oxidized. You lose electrons and a hydrogen atom that are transferred to NAD+. So NAD+ becomes reduced by creating a bond using the lost electron and hydrogen atom. I'm going to play that again.
Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
Okay, folks. All right, so I know this is kind of heavy stuff. And please ask questions if you have them. Sometimes it's hard to know what to even ask, isn't it? But let me ask you. Did that video help or hurt your understanding? Okay, all right. Okay, so folks, NADH is an electron carrier, okay, and NADH is a hydrogen atom carrier as well. Okay, so question from Afalashadi. Yes, Afalashadi, you can click this link in your notes and it'll bring that video back up for you, okay? So you'll be able to access it. Okay, so, all right, so we're getting a little bit into the weeds. Let's step back a little bit and start asking some questions to talk more about the big picture of things. Okay, so let's see here. Okay, Lance, could you please say hi to us?
Hello.
Hi, Lance. Please speak up nice and loud for us. Lance, let me just ask you a question here. Are you able to see figure 5.12 in your textbook?
Yes.
Okay, so Lance, my question is a general one for you. What is the purpose of the cell manufacturing NADH in the context of what we're talking about here in terms of aerobic cellular respiration? Why is the cell making this thing? What do you think, Lance?
I'm not sure.
Okay, it's an honest answer and I appreciate that. Okay, so thank you for trying. Just to move things along, let me answer this for everybody. Okay, the purpose of NADH is to act as a carrier of electrons and a carrier of hydrogen atoms, which will ultimately turn into hydrogen ions, to carry these electrons and hydrogen atoms to, drum roll, the electron transport chain. The electron transport chain cannot function without electrons. Where does it get electrons? It gets electrons from the electron carrier molecules that are made during glycolysis and later on in the Krebs cycle. That's why the cell is making this thing. So once again, the purpose of manufacturing NADH is to have a carrier of electrons and hydrogen atoms that can be used to energize the electron transport chain. The electron transport chain doesn't work without electrons and honestly hydrogen ions as well. So the cell is going through these chemical reactions of glycolysis and later the Krebs cycle to break down the nutrients in the foods that we eat and take into our body and rip some of those electrons and hydrogen atoms off those nutrient molecules in our food, put them onto NADH, which then acts as a Federal Express delivery guy to deliver those electrons to the electron transport chain. Because if I have electrons and hydrogen ions at the electron transport chain, I can use them to make ATP. Okay. All right. Thoughts, questions, folks? Okay. Let's continue. Question 10. All right, a lot of meaty topics, unfortunately, in this chapter. And I'm not asking you to memorize anything. I'm simply asking you to understand, which is the harder thing, isn't it? So, okay, let's continue here. All right, so I think up to bat is Rachel. Okay, Rachel, can you hear me okay?
Yep.
Okay, Rachel, here's my question for you. Okay, so Rachel, let's go back to chemical reaction number 1 there, okay, where you see glucose being converted into glucose 6-phosphate, okay? So Rachel, at chemical reaction number 1, when the phosphate atom is added to glucose, is that an example of oxidative phosphorylation, photophosphorylation, or substrate level phosphorylation?
I'm going to wait until you type that out so that I can remember all of the ones that you said.
Sure thing. And I want to just stop those beeps. That's my inbox getting lots of emails there. Okay, so at chemical reaction number 1, when phosphate is added to glucose, is this an example of oxidative phosphorylation, photophosphorylation, or substrate level phosphorylation? What do you say, Rachel?
Wow, they all look so similar. I want to say oxidative phosphorylation.
Okay, Rachel, is that your final answer?
That's what I'm gonna go with. That's what I'm gonna go with.
All right, thanks, Rachel. Appreciate your participation today. Unfortunately, if this were a game show, I would go, that's not right, okay? So this
You gave me all the big words.
I know, that's not fair, is it? Okay, all right. So your textbook goes over this, right? I mean, this is in your book. Take a look at, if you found figure 510 in your textbook, then you should see a section of text that says the generation of ATP, and it goes over substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. Okay, the big words here. And the point of it is that it's in your book. You need to think about it. You need to read it and see if you understand what it means. So in terms of adding phosphates, when you take a phosphate off a molecule and add it to another molecule, that's called phosphorylation. And when you take a phosphate off a molecule and link it onto a molecule, that is specifically substrate level phosphorylation. Okay, so the answer here is substrate level phosphorylation because we're ripping a phosphate atom off of ATP and gluing it to the end of glucose. That's why this is substrate level phosphorylation. Okay, and so let me say the answer to this question cannot be photophosphorylation because photophosphorylation involves sunlight, it involves light. That is a photosynthesis thing. We're not talking about photosynthesis here. Light is not needed in any way to make glycolysis happen. So photophosphorylation cannot be the correct answer. And oxidative phosphorylation, I will admit, is a little bit more of a sticky thing to understand. But basically, oxidative phosphorylation is something that takes place at the electron transport chain. And right now, in our conversation of aerobic cellular respiration, we're not talking about the electron transport chain. We're talking about glycolysis. So by process of deduction and by elimination, the only reasonable answer and the correct answer is substrate level phosphorylation. Okay. All right. Thoughts, questions, curse words? All right, let's keep going here. Let's keep going here. All right, so I think, I think I'm going to leave glycolysis at this point and move on to the next part of aerobic cellular respiration. Okay, because I'm looking at my clock here and we do have to move on. So my point here, folks, in giving you questions like this is to honestly, probably a little bit from my teacher perspective, gently scare you that you really need to study this stuff. It's not okay to not understand what a substrate is. It is not okay to not understand the purpose of NADH. That is not okay. You should be understanding these things as you process through this chapter. Okay, so glycolysis is the first group of reactions of aerobic cellular respiration. And we've asked you several questions about it in figure 512. Is there anything you'd like to ask about glycolysis before I leave it? The truth of the matter is now I'm going to start going a little bit faster, simply because we got to be sure we cover this content. But these questions are what I want from you. Samantha, did you have a question?
I just wanted to clarify something really quick. I'm pretty sure we already went over this, but I just wanted to clarify. Glycolysis produces ATP and then reduces the NAD+. Is that right?
So let's take each of those statements at one time, because they're good statements. So yes, glycolysis does manufacture ATP. And in fact, Samantha, let me answer your questions. Glycolysis makes ATP, true. And is NADH manufactured by reducing NAD+? That is true. Samantha, have I answered your two questions?
Yes, thank you.
Okay, so Samantha, stay on with me. Since you brought it up, I'm going to ask you the next question here. Okay. So Samantha, let me ask you this, and it was a good thing that you asked your question. Here we go. How many net ATP are made from the breakdown of one glucose molecule during glycolysis, Samantha?
It's either 2 or 4.
Okay, you got to give me a final answer. This is a game show.
I'm gonna go with 4.
Okay, thank you. No, it's not 4, it's 2. And key to that is this phrase right here, net ATP. That's the key, okay? So there are 2 net ATP made. Okay.
So then it's 2 net ATP is 2, and then it's 4 glucose molecules that produce the ATP. Is that correct?
No. So let's take a look at this. So stay with me, everybody, okay? So take a look at figure 512. Chemical reaction number 1, we actually break down an ATP molecule. We don't make an ATP, we break it down. Chemical reaction 3, we break down another ATP. So far in the glycolytic pathway, we have consumed 2 molecules of ATP and we've made none. When you get to chemical reaction 7, we make 2 ATP, but we've already consumed 2 ATP, so we make 2 ATP, so our net energy yield at that moment, at the end of chemical reaction 7 is 0, because we ate 2 ATP and then we made 2 ATP, so we're starting back at the starting point again. When you get to chemical reaction 10, you make another 2 ATP, which is why we're telling you, you manufacture 2 net ATP. You get 2 additional ATP out of the glycolytic pathway by actually consuming 2 ATP first, then making a total of 4 ATP. So 4 minus 2 is 2. So you make 2 additional ATP above and beyond what you had to consume to make glycolysis happen. Samantha, does that
Okay.
make sense?
Yeah, no, that makes sense. I was getting the 2 and the 4 confused, so, or like mixed up. So that makes sense. Thank you.
Okay, great. Thanks for asking. Any other questions? Okay, so folks, all I'm saying is that you do have to study this stuff, okay? Let's go on to a question and answer session, and the question and answer session will be for glycolysis that we see in figure 513. I'm sorry, Krebs cycle 513. And this won't be as extensive because, I do have to move on to some other things, but I promised everybody I'd give at least everybody 1 question, so that's what I'm going to do. Okay, so okay, so Tiffany, can you are you able to say hi to us, Tiffany? Tiffany, you out there? Are you able to unmute your microphone and say hi? Okay, I guess Tiffany might be a little preoccupied right now. We'll move on here. Okay. Oh, okay, Tiffany, you're having the same problem as someone else. I don't know what's causing that. I don't. So I'm sorry if you're currently locked out there. It's okay, Tiffany. We'll go on. Let's see here. Taylor, are you able to unmute and say hi?
Yeah, looks like it. Hey.
Okay, great. Okay, Taylor, great. Taylor, I'm going to ask you, with figure 513 in front of you, Taylor, here's my question to you. What molecule does the Krebs cycle start with?
Is that pyruvic acid?
Okay, so is that your final answer, Taylor?
Yes.
Okay, thanks for participating today. I appreciate it. Taylor, we're on a game show right now. Unfortunately, I got to do this. Okay, so take very careful look at this, folks. The Krebs cycle actually begins with acetyl-CoA. It does not begin with pyruvic acid. Okay, it begins with acetyl-CoA. Do you notice, Taylor, do you notice that blue ball number 1 is located at the acetyl-CoA? It's not located at the pyruvic acid.
Yeah, I thought it was a trick question. I was gonna say the second one.
Okay.
I went with my gut, which was the pyruvic
Yeah, it's
acid.
like if your teacher's asking it, it's probably a trick question, right? Okay, so thanks, Taylor. So what molecule does the Krebs cycle start with? It starts with acetyl-CoA. Okay, so question 13. Taylor, let me ask you another question, see if you can redeem yourself. Taylor, is acetyl-CoA the end product of glycolysis?
No.
Okay, good job. It isn't, right? I mean, if you just pan back to figure 512, you see the end product of glycolysis is pyruvic acid. So pyruvic acid is the end product of glycolysis, but Krebs cycle starts with acetyl-CoA. What the heck is going on here? Well, that's why I'm bringing this up, because we need to talk about it. Okay, good job, Taylor. Thanks. No, pyruvic acid is. So here's the deal. The end product of glycolysis cannot immediately be used to drive the Krebs cycle. There has to be an additional single chemical reaction take place to link glycolysis to the Krebs cycle. That single linking chemical reaction is that top chemical reaction of figure 513, where it shows pyruvic acid to acetyl-CoA. The conversion of pyruvic acid to acetyl-CoA is called the transition reaction in chemistry books. And the purpose of the transition reaction is to enable the end product of glycolysis to feed into the Krebs cycle. You have to convert the end product of glycolysis into the start product, into the starting material of the Krebs cycle. This is necessary to link glycolysis to the Krebs cycle. Okay, all right. So it looks here that I've given everybody, either last time or this time, I've given everybody a question, with the exception of some kind people that are having some technology issues right now. So at this point, folks, I'm going to move forward with some standard lecturing to bring up things that I would want you to pay attention to, okay? So let's continue here. We're going to continue our coverage of the Krebs cycle. So I'll put it this way: points to think about when studying Krebs. Number 1, Okay. The Krebs cycle is also called, it is also called the carboxylic acid cycle. And the Krebs cycle is also known as the tricarboxylic acid cycle. Okay. So there's alternative names for this group of chemical reactions. Okay. The Krebs cycle, you'll notice that, well, let me just ask everybody, we'll just get your feedback on this. Here we go. How many net ATP are made by the Krebs cycle per glucose? Okay. All right. So, whew. Yeah, thanks for everybody that's contributing. I appreciate it. And just look at the answers there. And let me say, I know students always think teachers are trying to trick them. I'm not trying to trick you, gang. Honestly, I'm not. But the answer is 2. It's not 1, it's not 36. Notice the question says, how many net ATP are made by the Krebs cycle per glucose? What you will notice in videos that I've already shown you and which you should review, that the Krebs cycle will actually spin around twice. So you go around once to make 1 ATP, you go around a second time to make a second ATP. And so each spin of the Krebs cycle gets you 1, but you need to spin through the Krebs cycle twice to produce the atoms of 1 glucose, to process the atoms of 1 glucose. So you get 2 net ATP made per individual glucose that you started way up with glycolysis. Because again, if you take a careful look at figure 512 of glycolysis, you'll notice that glucose at the top has 6 carbon atoms. And at chemical reaction 4, those 6 carbon atoms are split in half. So you end up with 2 3-carbon molecules. The 2 3-carbon molecules are used to make 2 pyruvic acids. So the 2 pyruvic acids originate from splitting the 1 6-carbon glucose molecule. Each of those 2 pyruvic acids has to spin through the Krebs cycle once to get 1 ATP. And so because you're making 2 pyruvic acids from 1 glucose, you need to spin the Krebs cycle twice to get 2 net ATP per glucose. Okay. All right, let's continue here. Next thing I would bring out in figure 513, notice that we do have some NADH molecules getting manufactured in the Krebs cycle, just like we did in glycolysis, we made some NADH. Notice that there's also a new molecule there called FADH2, okay? So the Krebs cycle makes 2 different types of electron carrying molecules. They are the NADH that you're used to from glycolysis, and they are the FADH2. Folks, I don't really want to get into the chemistry of those things right now, but let me just say this. You should think of NADH as the Federal Express delivery guy for electrons to the electron transport chain. and you should think of the FADH2 as an equivalent UPS delivery guy for electrons to the electron transport chain. Okay, so this is the FedEx guy. Delivering molecules to the electron transport chain. And this is the UPS guy delivering electrons to the electron transport chain. Okay. All right, folks. What else here? I think I'll mention one more thing and then we'll move on. And that is that notice that in the chemistry shown to you in figure 513, there are a couple chemical reactions which you actually manufacture CO2, right? So the transition reaction and way up at the top, notice that CO2 is getting manufactured. Notice that CO2 is also getting manufactured at chemical reactions 3 and 4. Okay, so the transition reaction before the Krebs cycle makes carbon dioxide, CO2, and the Krebs cycle itself makes additional CO2. So if you have ever wondered where the carbon dioxide comes from that you eventually exhale out of your lungs, well, this is where it's coming from. Every cell of your body that's performing the chemistry of aerobic cellular respiration because you dared to eat a cheeseburger is making carbon dioxide during the transition reaction and the Krebs cycle chemistry. That CO2 eventually percolates into the bloodstream and you deliver it attached to hemoglobin to the lungs and you exhale it out of the body, all right? So that CO2 waste gets out of the body and you make it during the chemistry of aerobic cellular respiration. Folks, thoughts or questions? Okay. Yasmin, I agree with you. This is a lot to process. It really is, you know. So it's just an unfortunate reality that we've all picked arguably one of the most complicated topics on the planet to actually make a career out of, right? Because it doesn't get much more complicated than biochemistry of cells, okay? You might say, well, maybe quantum physics is more complicated, and I might agree with you, but it doesn't get much more complicated than the chemistry of cells. Okay, so I'll say this, you know, I'll say this to you. When I was in graduate school, I had to take a year of biochemistry, and we probably spent a month on what I'm now covering in like 3 lectures with you. We spent a month on this stuff in grad school, and my professor, my biochem professor, he would walk into the room without any notes, without a textbook, with a single piece of chalk, and he would write out all those chemical reactions that I'm referencing to you in the book from memory on the board and talk about them. And so you might say to yourself, wow, that guy's really a geek. Maybe he was, I don't know. But to you and I and the everyday person on the street, you don't really care about this stuff because it's simply a way to pass a course to get a career started, okay? But let me tell you something. My chemistry professor that could do all this stuff off the top of my head, he was, at the moment he was lecturing to me, he had cancer. He was a cancer patient. And one thing you may or may not know is that cancer cells do this chemistry differently. Cancer cells exhibit altered glycolysis, altered Krebs cycle, altered electron transport chains. What I'm telling you is my chemistry professor was not only a chemistry researcher, he was a cancer patient, and he was trying to save his life because there were no options available to his cancer to successfully treat him. Okay? That's when it gets real. That's when you give a shit. Okay? So we need people that know that stuff, or we're never going to solve the cancer problem. So long story short, he would do all this stuff for me, and I was blessed to have him as a lecturer because he really knew his stuff. But I also say that before I graduated from grad school, he passed away from his cancer. It got him. Okay, it got him. So this is why we do this stuff, because someday somebody in your family is going to have cancer. God forbid, someday we are all going to have cancer. And we hope that somewhere there was a student that started paying attention to this stuff. Okay? So let's continue. Let's continue. The electron transport chain. Okay, the electron transport chain is described to you on figure 514 of your textbook. And it is other aspects of electron transport and the production of ATP are described in figure 516. Okay, so I think now is probably talking points about the electron transport chain and production of ATP. That brings up another point. Point number 5. No, okay, I'm good. Okay. So after we're done covering this, folks, I will give you some summary notes about these processes. Okay. Talking points about the electron transport chain and production of ATP. Okay. Number 1. And this is figure, I think what I'll do, figures 514 and figure 516. Okay. Figure 514 and figure 516 is the relevant topics right now. I'm going to show you a video about the electron transport chain, because I think that will help you when I'm talking about it, when I'm giving you my talking points. So the video I'm going to share with you is this one here. And if you go to YouTube and type in electron transport chain, you're going to get a lot of crazy videos. I mean, there's a lot of stuff out there, okay? So let's watch this as a way to start our conversation. I expect that we're going to probably need 15, 20 minutes of lab to finish this off, but that's okay. We can do that, okay? All right, folks, so let's watch this and see what it has to explain to us, okay? This is actually a pretty new video. Like some videos, it does have a little bit of cheesy music in the background. I apologize for that. Just ignore the music and see what this has to share with us. Here we go.
All of the steps of aerobic respiration that we have done so far have been leading up to this, the electron transport chain. This is the main event, the big bonanza, the Super Bowl of cellular respiration. Why? Because the vast majority of our ATP is made here. The electron transport chain is actually a series of proteins and other molecules embedded in the inner membrane of the mitochondrion. These molecules transport electrons, hence the name. Let's zoom in on this inner membrane to see the electron transport chain in more detail. Here's how it works. NADH, that electron carrier we've been making all along, delivers its electrons to the first protein in the electron transport chain. As the electrons move through this protein complex, energy is gradually released, and this energy is used to pump hydrogen ions, or protons, across the inner membrane, from the matrix into the intermembrane space. At this point, it might help to realize that moving electrons is electricity, so it should not be surprising that these electrons can provide the energy to drive these microscopic pumps made out of protein. They are basically natural electric pumps, created way before humans invented them, which is super cool. The The electrons are then transported by this special red molecule toward their next destination. The red molecule also picks up the electrons donated by FADH2, that other electron carrier that we made during the Krebs cycle. All of these electrons are then passed on to another protein complex. At this next protein complex, the electron energy is used once again to transport hydrogen ions across the membrane into the intermembrane space. Notice that we are building up a high concentration of hydrogen ions in the intermembrane space. This will be very important soon. Now the electrons are transported to a final protein complex, where their energy is used once again to pump hydrogen ions into the intermembrane space, making the concentration even higher. Now the electrons need somewhere to go, a final destination to complete their journey. Fortunately, you breathe, and oxygen comes to the rescue. Oxygen accepts the electrons, as well as hydrogens from the matrix, making water. This is the reason why you breathe all day, every day. Your cells need oxygen to accept these electrons, or the electron transport chain will grind to a halt, like an LA freeway during rush hour. And if the electron transport chain stops, you won't be able to make enough ATP to keep you alive, and you die. So keep breathing that oxygen. Now you may have noticed that we haven't made ATP yet, so you might be wondering how that works, which brings up the coolest part of this entire process in my opinion. Remember that high concentration of hydrogen ions we've been building up? Now it's time to use it to make ATP. A protein called ATP synthase makes a channel that allows hydrogen ions to flow through from high concentration to low concentration. You may remember that molecules tend to move from areas of high concentration to low concentration naturally, a process known as diffusion. So, these hydrogen ions, in a manner of speaking, want to flow from the high concentration area of the intermembrane space to the low concentration area, the matrix. But they can only get through ATP synthase. This facilitated diffusion of hydrogen ions through ATP synthase causes ATP synthase to spin like a turbine and produce ATP. Such a cool mechanism. It's like a microscopic hydroelectric dam, generating ATP instead of electricity as hydrogen ions passively flow through. Since we made many electron carriers throughout respiration, the electron transport chain happens over and over again, making between 30 to 34 ATP molecules for every glucose that entered respiration. If you are doing the respiration activity at Bioman Biology,
Okay, folks, so we can cut that off at this time. That is just one of many videos that you could watch. I don't have time today to show you this video, but I'm going to put the link in your notes. This video here gives you more detail about the motion of the ATP synthase enzyme, and it's only 3 minutes. I would encourage you to watch it during your private study time, not because you need to know it, but because it's really cool. It's really cool what we can figure out here. Okay. All right. So folks, it's basically 1:45 at this time. I need 10 or 15 minutes to finish this topic up in lab, and we'll do that. So at this moment, I'll pause what I'm doing. Hi, folks. Okay.
Hello.
Let's see what we can do here. We'll try our best to finish up chapter 5. I might give you just a little bit of information from chapter 6, and then we'll get lab started today. So hang in there. Okay, let's continue with our conversation. So last time, we had just finished showing you the video on electron transport. And so now I just want to give you a couple observations or points that you should think about while you're studying this topic. Okay. First off, number one, one way to think about the electron transport chain, if you look at figure 5.14, look at figure 5.14. One way to think about the electron transport chain is as an analogy, the electron transport chain is similar to a staircase in the sense that if you look at figure 5.14, notice you have that molecule there that's called FMN, that's flavin mononucleotide, okay? You don't have to know that, but FMN. And you notice that you go down to Q, you go down to cytochrome B, cytochrome C1, so on and so forth. So if we'd had a tennis ball at the top of a staircase and we pushed it off the top step, it would roll down each subsequent step. So the electron transport chain, from one analogous perspective, can be thought of an electron staircase, where we're moving electrons from one step of the staircase to the next step of the staircase to the next step of the staircase. And the whole purpose of doing that is, in fact, to push hydrogen ions to the other side of the membrane. Okay, so a couple ideas that come out here. Okay, remember that when I say electron transport chain, I'm just going to abbreviate that ETC to make it shorter, okay? ETC is the electron transport chain. Remember, the ETC takes place in eukaryotic cells in the inner mitochondrial membrane, okay? That's the physical location where this is taking place. And remember that the ETC takes place in the plasma membrane of prokaryotic cells, right? Because bacteria, as an example of prokaryotic cells, they do not have mitochondria, but they do have a plasma membrane. The plasma membrane is where all the chemistry of the electron transport chain happens in bacterial cells. The corresponding location of where the electron transport chain happens in eukaryotic cells is the inner membrane of the mitochondria. Of course, you remember from exam 1 that the endosymbiotic theory suggests that, in fact, mitochondria really are primitive forms of bacteria. Okay. All right. So that's the first thing. The electron transport chain takes place in the inner mitochondrial membrane of a eukaryotic cell, like our own human cells. And in bacteria, it happens in the plasma membrane. You can think of the ETC in either a prokaryotic or eukaryotic cell. You can think of the ETC as a staircase where electrons start at the top step and they progressively migrate to each lower step in the staircase. Each lower step of the staircase are going to be all those molecules that are lightly shaded purple in figure 5.14. They're also lightly shaded purple in figure 5.16. So you can think of the ETC as a staircase where electrons move down each step of the staircase. And as they do, as the electrons move from one purple molecule to the next purple molecule to the next purple molecule, there's going to be several moments in time where hydrogen ions are pumped from one side of the membrane to the other side of the membrane, okay? And as they do, hydrogen ions are pumped across the membrane, okay? And so think about it. You all know, I know you know, if you think about it for a moment, you know what the source of hydrogen ions are. And if you don't know, you can look at figure 5.16 quickly and you say, oh, there's NADH, right? Figure 516, orange ball number 1, do you see NADH? So NADH is being oxidized to NAD+. When NADH is oxidized, you rip electrons off, which you see are color-coded red in figure 516. You rip electrons off NADH and you push hydrogen ions through that protein to the other side of the membrane. Okay. Remember that electrons that energize the ETC are provided by oxidizing the electron carrying molecules, NADH and FADH2. So you oxidize these molecules, you break bonds, you pull off a hydrogen atom. When you strip the electron away from it, the hydrogen atom becomes a hydrogen ion. You push the electrons and the hydrogen ion into FMN, and the electrons begin their journey down the staircase, and the hydrogen ion gets pushed to the other side of the membrane. Okay. All right. Thoughts or questions? So the oxidation of the electron carrying molecules is going to happen at several steps of this chain, and you're going to push hydrogen ions across. The whole point of pushing hydrogen ions across to the other side of the membrane is to intentionally create a hydrogen ion gradient, where you have a whole bunch of hydrogen ions on one side of the membrane and very few hydrogen ions on the inner surface of that membrane. That's the whole point. Okay, I keep saying the other side of the membrane. Well, that membrane is going to be the plasma membrane in bacteria, and it's going to be the inner mitochondrial membrane in a eukaryotic cell, right? So keep that in mind. So the whole purpose of pumping hydrogen ions to the other side of the membrane is to intentionally create a gradient. Okay, so you might ask yourself, why? Why do we need a hydrogen ion gradient? We need a hydrogen ion gradient, but we need a hydrogen ion gradient because Mother Nature, in her infinite wisdom, engineered a certain process that has to be followed to manufacture ATP. And you could ask yourself, well, why is it done this way? Why didn't Mother Nature create an alternative solution? And I suppose that's a good philosophical question, but it's not one that we can answer. Mother Nature created what she created. We observe it, we measure it, we report on it, so we got to understand it. Why do we need a hydrogen ion gradient? Because the machinery of the cell evolved. Cells evolved machinery to use the motion of hydrogen ions back across the membrane to actually manufacture ATP. Okay, in the video, I think the narrator mentioned like a hydroelectric dam. If you know anything about a dam, right, you've got water at the top. It flows over the dam and it falls down. And when the water flows down, it hits certain machinery at the bottom. It spins a turbine to generate an electrical current, to generate electricity. It's the same idea here. We've got to produce a hydrogen ion gradient so there's water at the top of our cliff. We're pushing the hydrogen ions to the top of our cliff, like water at the top of a cliff. And then we let the hydrogen ions flow down the cliff, down back through the membrane, so we can actually make electricity. We can make ATP. So why do we need a hydrogen ion gradient? To allow the cell to manufacture ATP when hydrogen ions flow back across the membrane through a special enzyme called ATP synthase. Okay. All right. All right, folks. So let's start wrapping this beast up here. Keep in mind that in prokaryotes, the electron transport chain, well, let me ask you this. Let me ask you something in the chat window, okay? Because I think you have some background you can draw upon and contrast with information that I'll be sharing with you. Hold on a sec. How many total net ATP are made from the breakdown of one glucose molecule during all of the chemistry of aerobic cellular respiration? What have you learned in other courses? Oh, boy. Take a look at your answers, folks. Take a look at that. It seems like there's some disagreement. Okay. All right. So here's the deal. The answer to that question depends on knowing the identity of the cell that's performing the aerobic cellular respiration. In a human cell, which we'll use as a typical eukaryotic cell, you will get 36 net ATP by breaking down one glucose molecule through all of the chemistry of glycolysis, Krebs cycle, and electron transport chain. If we are talking about a bacterial cell, a prokaryotic cell, you will make 38 net ATP. Okay, so the answer to that question depends on knowing the identity of the cell. Remember, eukaryotic cells make 36 net ATP per glucose. Hold on a second. Let me define this differently for you, okay? Because I don't want to confuse yourself when I just kind of confused me. We're talking about the electron transport chain. Eukaryotic cells in the electron transport chain, not all of respiration, will manufacture, and let me get my notes so I don't mess this up for you, 32 net ATP. Remember, when considering the total ATP made by the ETC, we get eukaryotic cells make 32 net ATP per glucose. That is simply for electron transport. Prokaryotic cells make 34 net ATP per glucose. That is for just the electron transport chain portion of this. Okay. So now let's summarize everything. Okay, so we've talked a lot. We've asked a lot of questions. We've tried to answer many of them. We have shown you some videos. And I will remind you, if you don't remember, that your textbook does have a source of questions, right? At the end of each chapter, there are questions that you could attempt to answer during your study time to get a better idea of whether you're really grasping this content, right? Study questions in my physical book are page 148, 149, 150. All right, so the summary of all aerobic cellular respiration, we'll break it down this way. Okay, I'll give you a 3-column chart. think we can do this. Okay, so we're going to make a statement here. Then we will answer that statement for prokaryotic cells. And eukaryotic cells. Okay. All right, so the first thing is we'll mention glycolysis. Wow, why does it keep doing that? All right. All right, so glycolysis in prokaryotic cells takes place in the cytoplasm of the cell. It takes place in the cytoplasm, and you manufacture 2 net ATP per glucose. For glycolysis in eukaryotic cells, you have exactly the same scenario. The chemistry takes place in the cytoplasm, and you make 2 net ATP per glucose. Okay. Now, I will put down the transition reaction here. Okay, for the transition reaction, that is going to take place in a prokaryotic cell. It's going to take place in a cytoplasm. Okay. You do not make any energy here. The transition reaction is not about making energy. The transition reaction is about converting the end product of glycolysis to the starting material for the Krebs cycle. So no ATP made. No ATP made. Okay, in eukaryotic cells, the transition reaction actually occurs across the outer mitochondrial membrane, because the transition reaction is moving the chemistry of the Krebs cycle into the mitochondria. Across the outer mitochondrial membrane. And again, there's no ATP made by this. Next, we have the Krebs cycle. The Krebs cycle in a prokaryotic cell, right? Prokaryotic cells don't have mitochondria. They generally don't have organelles per se. So Krebs cycle is going to take place in the cytoplasm. And with the Krebs cycle, you're going to generate an additional 2 net ATP per glucose. With eukaryotic cells, the Krebs cycle takes place in the mitochondrion, because the transition reaction chemistry happened across the dimensions of the outer mitochondrial membrane. Mitochondria, and we make 2 net ATP per glucose here. Okay, looks like I'm going to need one more row in my table. All right. Then we have the electron transport chain, the ETC, which we just covered, right? The electron transport chain in prokaryotic cells actually happens across the plasma membrane. And because it's a prokaryotic cell, you're going to get 34 net ATP per glucose. In eukaryotic cells, the chemistry of the transition, the chemistry of the electron transport chain will happen across the inner mitochondrial membrane. And for a eukaryotic cell, you will get 32 net ATP per glucose. Okay, so the energy yield of these processes is identical between prokaryotic and eukaryotic cells for glycolysis and the Krebs cycle, But when we get to the electron transport chain, there is a 2 ATP difference. So 34 net ATP in the ETC of prokaryotic cells, 32 net ATP in eukaryotic cells. So our grand tally here for ATP is as follows. Okay, prokaryotic cells, you get a grand total of 38 net ATP per glucose. And eukaryotic cells, you get a grand total of 36 net ATP per glucose. All right, gang. All right, so there's always more that can be done, blah, 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 right? There's always more that can be said, but I feel pretty decent about this. So would you like to ask any questions about this material that we've talked about? Is there anything on your mind you'd like to ask about? Okay. All right. Then I don't want to spend a lot of time in chapter 6. I had hoped to start it today, but here's what I'll do. For the benefit of those that might be interested in dangling their feet into the water on that on the workbook problem, that math problem, I'm going to give you a couple equations in your notes at this time, which you'll need to actually wrestle with it. So if you want to make the attempt over the weekend, you can. You don't have to, but you can, okay? All right, so let me start a new document for chapter 6. Chapter 6, and chapter 6 is the final chapter worth of content that will ultimately be on exam 3 when you come back from Thanksgiving, okay? So let's just give you some equations here that you'll need to tackle here. Chapter 6, the topic of chapter 6 is microbial growth. Okay. And so there really isn't much to this, but I'll send this along today and we'll continue in chapter 6 next time. But okay. So microbial growth formulas for workbook number 4 assignment. Okay. So folks, I'm going to put 3 equations into your notes. And here's the deal. I'm giving you 3 equations. I'm not telling you that you must use all 3 equations in your homework. I'm not telling you that. I'm saying here's three equations. You may or may not need to use all 3 equations to get the answers to the workbook assignment correct. Okay? So you need to know when to apply which formula. Sometimes you may not have to use all of them. Sometimes you might have to use all of them. Okay. So here's formula number 1. Here's it. If we want to calculate the total number of cells that are present in a population after some period of growth, right? If we started out with 1,000 bacterial cells and we allowed them to grow for 10 hours, how many bacteria would we end up with? Where there are ways to deal with this. And that's part of what we're going to talk about in chapter 6 next week. So right now, I'm not going to lecture on it, just going to give you the equations, okay? So the final number of cells is equal to the starting number of cells multiplied, okay? Multiplied, that's an X there for multiplication, multiplied by the number 2 raised to the power, that caret symbol there, that unusual looking upside down V is called a caret symbol in computer science. 2 raised to the power of the number of generations. So the final number of cells equals the starting number of cells multiplied by 2 raised to the power of the number of generations, okay? So to make sense of that, we have to think about, well, what do we mean by number of generations? Okay. The number of generations equals the number of times the population of cells has had the opportunity to reproduce and double themselves, okay? So the number of generations equals the number of reproductive cycles the population of cells have gone through. So I have a question for you in the chat window. I have a question for you in the chat window. Here we go. If we start with 1 cell and finish with 8 cells, how many times did how many reproductive events must have occurred to get 8 cells? If we start with 1 cell and finish with 8 cells, how many times did reproduction occur to get the 8 cells? Thank you, Yasmin. How about the rest of you? Okay, all right. So thank you for answering. That shows me that I do need to spend some time on it. The answer is 3. The answer is 3. Okay, so let's get this into the notes. If we start with 1 cell and finish with 8 cells, how many reproductive cycles have occurred to get 8 cells? Here's the deal, folks. Think about it. One cell goes through binary fission to produce 2 cells. 2 cells go through binary fission to produce 4 cells. 4 cells go through binary division to produce 8 cells. So each arrow in my flow chart represents a binary fission event where the cells that exist at that moment are reproducing themselves to double their number. 1 cell doubled to 2. That's 1 reproductive event. That's 1 generation. The 2 cells reproduce to get 4. That's a 2nd reproductive cycle. The 4 cells then reproduce to get 8. So all told, for 1 cell to grow into a population of 8 cells, we must have gone through 3 generations, 3 reproductive cycles. Okay. Each arrow is a binary fission event. Okay. Each arrow is a generation. Okay, so we have 3 generations between 1 starting cell and 8 final cells. Okay. All right, folks, let me get a feedback from you. Is this making sense? The way that I've presented it, does it make sense now? Okay. All right, so once you get used to that kind of logic, things go more smoothly, okay? All right, so that's the first formula. Here's the next formula. We do, there are times when it will be obvious what the number of generations is. There'll be times when the can't quite highlight this the way I want. Okay. there will be moments where the number of generations will be provided in a problem and you'll just know it. There'll be other times when you got to calculate it or reason it out. So here's an equation that if you're given certain information will allow you to calculate the number of generations, okay? Number of generations is going to be equal to the following. And this is where your ability to use the log key on your calculator comes into play, okay? I'll cover this later with you again next week, but just to bring up my calculator here, okay? Just to bring up my calculator here, right? Here's my log key, LOG, that's base 10, right? So in my calculator, I can type in 1000, hit that log key, and I should get the number 3, and I do. If you can do that on your personal calculator at your house, you're good. You're golden. That's all you need to be able to do for these math problems, okay? So the number of generations equals the log of the number of cells at the end of growth minus, that's a minus sign there, minus the log of the number of cells at the start of growth, all divided, okay, so I'm going to underline this, all divided by the following decimal, 0.3010299, okay? And what I'll show you is this. That might seem like an arbitrary decimal here. It's not so arbitrary. Watch what I do in the calculator. Okay, so I'm going to clear out that number here, clear the entry. I'm going to type in the number 2, and I'm going to hit the log button. Look what I got. 0.3010299. 0.3010299 is the logarithmic value of 2. That's why it's in this equation, because every cell divides to 2. Okay. So that's the second formula. The third formula is this, formula 3. There may be some problems, you know, and obviously right now this is school, right? So every problem I give you is academic because nobody cares about school. But in the real world, you know, for companies that manufacture beer and things like this, which we probably all enjoy occasionally, you have to be able to do these kinds of calculations when you're manufacturing a biological product. Okay. Okay. Thank you, Rachel. I appreciate that. Okay. So formula number 3 is about calculating the generation time. Okay. So first off, generation time is the amount of time it takes for a population of cells to go through one round of reproduction, one round of binary fission. Okay, and so with certain information, you can actually calculate this, and I'll break this down a little bit further for you. So generation time equals the following. It equals 60 minutes per hour, 60 minutes per hour, multiplied by the number of hours the cells have been growing, all divided by the following, all divided by the number of generations that occurred during that period of growth. Okay. So these 3 formulas are now in your notes. These 3 formulas are in your notes. They are also in Appendix B as in boy in the back of your textbook, which is probably not a bad idea for some of you folks to read through. And we will be applying these formulas with practice problems next week and before Thanksgiving, okay? All right. Okay. All right. Any final thoughts or questions before we pause this material? Okay. So give it the old college try, folks. I know you can do it. I know you're all capable of doing more than you think you can do. Okay. Most human beings are. Okay. All right. So let's continue here. And I'm going to stop recording at this time. Okay.